Okay. So this paper is a bit of context that'll help you in interpreting the results from all the other vaccine papers that we've been covering. So this is the title of it. It's in the last folder along with the Moderna vaccine. And let's jump past. Well, actually, no, let's not jump right past it. But here we're looking at the endpoint titer. So think of it as the total amount. And so 4 means 10 to the fourth. That's why I'm saying 10 to the x over here. And so 10 to the fourth would be equivalent to 10,000. And so that's about the number we're hitting here. And the low end seems to be about 10 to the third, which is 1,000. So there you go. And you can look at all of those things. And then neutralization. Here we go. So these bluish lines are all different people. And so they're showing the amount. And so it looks like in this particular one with their version of the test, neutralization seems to be mostly about 256. These are people that made lower responses. They put them in order from low response to high response here. At the high end, you got about 512. There are other people with other versions of the neutralization test, because remember, everybody does it a little bit differently, crazy as that is, that actually come up with potentially different numbers for the same serum. So, yeah, grain of salt, but this is where we are with this version. Okay, and then this is going to go through the B cell and T cell responses, and there are a couple of things that are going to make sense here. So what they're doing over here, so this is that flow cytometry where they sort out cells according to whether or not it has certain markers on the surface. So here's one marker here, here's another marker, and if it has both markers, it's going to end up in this box right in the middle, just like those guys. And so CD19, you got to know what some of these markers are. Then all of this becomes suddenly interpretable. So CD19 is a marker for any kind of normal B cell should have this. So these are things with the potential to produce antibodies, but they could be really early stage or late stage. It's hard to say. So how do we tell the difference? Well, we can look and see whether they are IgD and IgM positive or negative. And in this case, they're just going with the IgD because it's usually going to be positive for D and M at the same time. And in this case, if it is D negative, that means we've got ourselves a B cell that has gone past the initial stage. So it's either going to be making IgG or IgA, most likely, possibly also IgE, although that won't do us much good. But it's going to be like a mature B cell. That's what we're going for in this blue box here that's kind of at an angle. And down here, they're actually going a step further, saying, well, okay, yes, CD19, so it's a B cell. No IgD, so it's not a baby B cell. And then IgG positive, which means that, yeah, it's a non-baby B cell that's actually making proper IgG. And so that's what they are looking for there. And then they're saying, does it bind to the receptor binding domain? Does it bind to something else? And they're looking at it that way. Over here, these are looking at T cells, so it's T helper cells, and there are some T helper cells that help in the follicle region. So these are going to be involved directly in educating other immune cells and supporting them, let's say. And so that's what the CTFH stands for, I believe. Of those, you have a couple of markers here. So the first one is going to be CD3 positive. All your, let's say, white blood cells, all your T cells, helper and killer, whatever, are going to be CD3 positive. CD4 positive is going to tell you that these are cells that have the marker that sets T cells, rather helper T cells, apart from the others. So CD3 says could be a T cell. CD4 says, yep, it's a helper T cell. CD45 is a marker of memory T cells, CD45RA. And so if these are negative for that, that means they're not yet memory cells. So they're fairly recent, not from something that you caught while you were a kid. And then CXCR5 is going to be a T cell, kind of a homing marker. And this is taken by most people as the marker that they are dealing with these follicular T helper cells in particular. And so they're trying to narrow, sort of zoom in on a particular population. And so when they're showing percentages here, and 1%, 2%, whatever, this is percent of cells that are both positive for CD134, which is going to be another marker that is often found on T cells, and positive for CD25, which is a different marker that is going to be found on T cells. And so between these things, they're narrowing it down, which is part of why they get higher percentages. But at the same time, it's also a little more precise. So it's hard to directly compare these numbers between this assay and the assays in some of the other papers. But at least you can see kind of a general trend and what's going on and what we should expect. Yeah, so that is the big thing here, is looking at those helper T cells. And then I can't remember if they looked at killer T cells in this paper or if those show up like in the supplementary information. I think they did measure those, because I think, let's see, follicular helper T cells. No, it looks like we just have a bunch of those, and B cells. So we're focused on the antibody response and support for the antibody response for the most part in this paper. So it's not perfect, but it's got a lot of good stuff. And at least it's some context. This is the problem. You're going to get all these details from the vaccine papers, but it is very difficult to have context. And I guess you have to take your context from the convalescent patient data that they include in those tests. But remember, these may be people that have had mild COVID or severe COVID, and you got to read the details, because, yeah, that's where the little devils are. So thanks very much. This is our context paper that you're welcome to use and cite in your critique of the last three.